Hello, welcome once again. In this lecture, we are going to study uses of alpha amino acids in various fields, then different reactions of alpha amino acid. Whenever uses of alpha amino acids are considered, no doubt, amino acids are very important building block to develop protein naturally in living cell, plants, animals, and various kinds of flora and fauna. So whenever the uses of alpha amino acids are considered, the most important use of alpha amino acid is protein synthesis. No doubt, all of us are familiar with importance of protein as a food material. So, alpha amino acids are key precursors of various types of proteins. They also build peptide bondage in proteins to build various kinds of enzymes. Enzymes we are also going to study in detail in next chapter. That is enzymes, protein and alpha amino acid combination gives formation of enzyme as well as hormones via regular type of peptide bonding. So alpha amino acids are important building blocks to synthesize protein. Second, these are also precursors of biomolecules apart from protein. Now, which particular biomolecules or proteins? That is, important biological compounds such as serotonin, in which tryptophan is key precursor, which is alpha amino acid. Melatonin and serotonin developed by using tryptophan as amino acid. Tyrosine is one of the key amino acid to develop synthesize dopamine and adrenaline. See, both dopamine and adrenaline are very important enzymes, proteins which developed from tyrosine, which is one of the alpha amino acid, and adrenaline is acting as a thyroid hormone. So, serotonin, melatonin, dopamine, and adrenaline is totally synthesized by using alpha amino acids such as tryptophan, tyrosine as a key precursor. So, alpha amino acids are used as a precursors to synthesize such a important biomolecules. This is the second use. Third very important use, that is the nutritional uses. No doubt, food materials, vitamins, proteins, various kinds of fruits are enriched with alpha amino acids and various types of proteins. So, dietary supplements and key metabolism of human body, living bodies, are totally accompanied by alpha amino acids. So, nutritional values are more in alpha amino acids, and essential alpha amino acids in the body They are not developed in body, but they are synthesized by various living organisms, our ecosystem, green planet, and various vegetables. So, alpha amino acids are having a good nutritional values. Very important. This is nutritional use. The next important use is the medicinal use of alpha amino acid. Now, what is the medicinal use? Most of the therapeutic purposes and pharmaceutical purposes are completed by alpha amino acid. For example, cysteine is one of the mucolytic agent. The amino acid solutions for patients is provided. The solution is made from cysteine, and cysteine is acting as one of the mucolytic agent. So such kind of medical use are carried out by the various pharmaceutical practitioners, and alpha amino acids also plays an important role. Then very fast and important use, that is the industrial use of alpha amino acid. No doubt industrial use, various kinds of food industries, beverages, then various types of bakery products, then various types of itineraries are use amino acids as a intermediate compound. Then aspartic acid, phenylalanine, glycine, buffer, flavoring agents, all such kinds of alpha amino acids are used in various types of food industries. Most of the cosmetics, beautifying agents or beautification products, that is cosmetics, are prepared, synthesized by using aspartic acid, glycerin. Glycine is acting as a buffer solution. Phenylalanine is a key ingredient of skin product, and hence it is used in a cosmetic and allied material. So food industries, cosmetic industries, and all allied materials are developed by using alpha amino acid. Now research uses, no doubt most of the alpha amino acids are used in a laboratory for biochemical studies, laboratory sequencing, peptide synthesis, chromatography, electrophoresis, all such kinds of purposes, experiments, research activities are done with the help of alpha amino acids. So, alpha amino acids are having such majority 6 various areas in which they are continuously used. That is importantly for protein synthesis, precursors of biomolecules, nutritional uses, medicinal uses, industrial uses, and various kinds of research activities can be done with the help of alpha amino acid. So these are the uses of alpha amino acid. Now we are moving towards a various types of reaction which is performed by alpha amino acid. Now the most important reactions, that is, first one is the acid base reaction. Okay, no doubt alpha amino acid is containing both groups as it is amphoteric in nature, that is NH2 group is itself as a base, COOH group itself as a acid. That's why alpha amino acids are having tendency to react with both acid as well as base. Whenever alpha amino acid reacts with simple HCl, it gives formation of quaternary ammonium salt, that is NH3 group, which is having positively charged, and it is our now quaternary ammonium salt. So reaction with acid, alpha amino acid gives formation of quaternary ammonium salt with net positively charged species, while the COH group present in the alpha amino acid is itself acting as acid. When it reacts with the base, no doubt it gives formation of sodium salt of alpha amino acid. In both reactions, first reaction does not give elimination of water, but second reaction, this reaction gives elimination of H2, water molecule. So formation of salt and water takes place. So simply, acid-base reactions, both NH2 group and COH group is given, and it gives formation of the regular reaction as a regular acid as well as base. So second reaction, important one, that is esterification reaction. Particularly, esterification reaction is given by carboxylic acid group present in the alpha amino acid whenever alpha amino acid reacts with alcohol in an acidic condition. Whenever in presence of acid, simple regular alcohol reacts with carboxylic alpha amino acid, it gives formation of ester by elimination of water molecule. So this is simple reaction, that is esterification takes place along carboxylic acid group with alcoholic group as a reactant in presence of acidic conditions. So this is the esterification of alpha amino acid reaction, particularly shown by COH group present in the alpha amino acid. So esterification is the simple reaction which is carried out in between COH group and alcoholic group. This is esterification. Second, reaction with ninhydrin. Particularly, ninhydrin reaction is given by amino group present in the alpha amino acid, and this is very important reaction. Alpha amino acid reacts with ninhydrin agent to give a deep blue or purple color. This is the test of alpha amino acid as well as protein also. So ninhydrin test is given by which group present in the alpha amino acid, that is amino group. Esterification reaction is given by the COH group, carboxylic acid group present in the molecule, that is esterification. So ninhydrin test is given by alpha amino acid. This is particularly test of NH2 group, and during the test deep blue or purple color is developed. So this is the ninhydrin test. The test aldehyde purple red color. The next reaction is the decarboxylation. Decarboxylation means removal of CO2 group from COH group of alpha amino acid, and this reaction takes place under heat or enzyme catalyst. So decarboxylation reaction is given by alpha amino acid, and in this reaction COH group of alpha amino acid reacts in presence of either heat energy or enzyme catalyst, and it gives formation of elimination of CO2, so that alpha amino acid is get converted into a simple amine. See the reaction. Alpha amino acid after heating gives formation of regular amine, and there occurs elimination of CO2 group. Means, for example, histidine, one of the alpha amino acid, Whenever heated in presence of enzyme catalyst, histidine alpha amino acid is converted into histamine, one of the amine. Similarly, tyrosine gives decarboxylation reaction. Whenever heated, it gives formation of tyramine as a regular amines with elimination of CO2. So, decarboxylation reaction is given by alpha amino acid in presence of heat or catalyst. So, this is decarboxylation of alpha amino acid. Decarboxylation. Now, the important reaction, deamination reaction. Deamination reaction, as per the name, deamination means removal of NH2 group from amino acid. That is, whenever any amino acid reacts with enzyme, that is, enzyme is acting as a catalyst. Deaminase is one kind of enzyme which is used to convert amino acid into a simple type of alpha keto acid. See the reaction, alpha keto acid. By elimination of ammonia gas, it gives formation of deamination reaction. So deamination is the reaction in which alpha amino acid eliminates NH2 group so as to form alpha keto carboxylic acid, and evolution of ammonia takes place. This is simply called as deamination reaction. Deamination, esterification, decarboxylation. All these reactions are given by alpha amino acid either by NH2 group or COOH. Okay, so this is the reaction shown by deamination shown by NH2 group. The next reaction is peptide bond formation. It is a regular reaction and very important. Peptide bond. What is peptide bond? Peptide bond is one of the linkage in which there is a CONH group is developed, and this CONH group is developed from two amino acid. Carboxylic acid of one amino acid combines with NH2 group of second amino acid, gives formation of CONH group, that is peptide group linkage in the longest molecule known as peptide bond formation. See the reaction. R1 first amino acid, R2 second amino acid, COH group of R1 amino acid combines with NH2 group of R2 amino acid so as to give formation of CONH group in the new protein. This is peptide linkage is the presence of protein, and such type of polypeptide linkage gives enlarged polypeptide molecule known as a protein. So peptide bond formation is nothing but it is the reaction in which longest chain of protein is developed from alpha amino acid known as peptide bond formation reaction. So see the reaction, this is the important reaction, peptide bond formation. So this is sixth reaction. Now seventh, oxidation of cysteine. Oxidation of cysteine. Cysteine is the alpha amino acid containing sulfur group, disulfide bond, and it is sulfur type of alpha amino acid. We have seen in classification. Classification shows the R group containing sulfur, it is containing disulfide bond, and cysteine is the sulfur containing alpha amino acid. Whenever its oxidation takes place, cysteine is get decomposed and cysteine is get converted into cysteine. Cysteine, one of the amine, and there is a elimination of hydrogen and electrons. So it is oxidation of cysteine, cysteine into cysteine, that is amine. So this is overall about oxidation of cysteine. So in summary we can see that we have seen here about 9 different reactions. First one is acid-base reaction. The groups involved are COH and NH2 both. Product is zwitterine and salts. In esterification, COH group is involved. Product is ester. Neon hydrogen test, NH2 group is involved. Purple blue color is developed. Decarboxylation, COH group is involved. Amine and CO2 is developed. Deamination, NH2 group is involved. Product is ortho-keto acid plus NH3. Peptide bond, peptide or protein is developed. COH and NH2 both are involved, and oxidation of cysteine gives removal of SH and it gives formation of cysteine. So these are the some of the reactions, simple reaction but continuously takes place in amino acids, so that esterification, acid-base reactions, decarboxylation, deamination takes place and different products are observed. So these are the some of the reactions which are shown by various types of alpha amino acid. Okay, thank you. Uh, Listen the video carefully, see all types of slides, and these reactions are very easy to understand. Okay, thank you.